Hello students, this lecture is for the program Bachelor of Science 3rd year, subject Botany, semester 5, course code BOD 101, course title Plant Tissue Culture. Title of the unit, Unit 4, Types of Cultures, name of the module, Testing Viability of Cells. I am Ms. Marsha Rodriguez, Assistant Professor in Botany from Carmel College of Arts, Science and Commerce for Women. In this module, you will learn the importance of testing cell viability, methods for testing viability of cells. At the end of this module, you will be able to understand the importance of cell viability test, and you will be able to explain the methods for testing the viability of cells. First, let us see the need for testing viability of cells. Cell death may occur in a cell suspension culture due to several factors. Viability or the ability of the cell to survive after isolation and during culture is very important. Cell wall formation, cell division, callus formation and growth of the culture are largely dependent on the viability of the cells. Now, let us look at the different methods for testing viability of cells. First is the cytoplasmic streaming. Next, reduction of tetrazolium salts. Third, fluorescent diacetate method. Next, Evans blue staining. Then we have calcoflow white staining, and lastly phenosephrinin staining. We will now study about cytoplasmic streaming as a method for testing the viability of the cells. Streaming of the cytoplasm and the presence of a healthy nucleus indicate that cells are viable. The cells are observed under phase contrast microscope since it is difficult to observe these aspects in unstained cells under a bright field. Next is reduction of tetrazolium salts. This test is used to measure the respiratory efficiency of cells by reduction of 2,3,5 triphenyl tetrazolium chloride to the red dye formazole. Formazole can be extracted and measured by spectrophotometer. Next is the fluorescent diacetate method, also known as the FDA method. It is the most frequently used staining method for assessing cell viability. FDA dissolved in 5 mg per ml of acetone is used. This solution is added to the cell suspension at a concentration of 0.01% and incubated for 5 minutes. When examined under a fluorescence microscope, viable cells fluoresce green. Non-viable or dead cells fluoresce red. Next is the Evans blue staining. Evans blue at a concentration of 0.01% is used. It is specific for dead cells. The stain is mixed with the cell suspension. The non-viable cells stain blue, whereas the viable cells remain unstained when observed under light microscope. Here is a diagram depicting Evans blue staining method. This is the cell suspension to which Evans blue stain is added. And then you can see when observed under light microscope that the non-viable cells take up the blue stain, whereas the viable cells remain unstained. Next is the Calcoflow white staining. Calcoflow white stain is taken up by cell wall. Calcoflow white binds to the beta-linked glucosides in the cell wall, which is observed as a ring of fluorescence around the plasma membrane. Next is the phenosafrinin staining. Phenosafrinin is used at a concentration of 0.01%. It is specific for dead cells. The viable cells remain unstained, whereas the dead cells stain red. So students, in this module, we have learned that cell viability after isolation and during culture is very important as the growth of the culture is dependent on the viability of the cell. The various methods for testing the cell viability are cytoplasmic streaming, reduction of tetrazolium salts, fluorescent diacetate staining, phenosafrinin staining, Calcoflow white staining, and Evans blue staining. These are the references for this module. Thank you, students.